transition metals work actually turns out to be very important for biology. Uh, and so transition metals are used in biology, usually in very small amounts, but for essential functions. And so the aspects of electronic structure, reactivity, the things you've recently learned about are all very important parts of certain biological processes. And today we're going to take a look at a few of those. So your textbook actually does not cover this topic. So I've uploaded a chapter from another inorganic textbook, which you can read through. We won't cover everything in there, but it does have good information on this topic. So as you probably know, not all elements are essential to life. This is a periodic table just showing which ones are known to be essential or thought to be essential, particularly for human life. And of interest to us, of course, is in this class are these transition elements. And you'll note that the ones that are thought to be essential are mostly first row transition metals. So vanadium through zinc, and then also molybdenum. And so these metals and their electron transfer and reaction chemistry are particularly important for certain biological transformations. For non-human life, there can be some different elements that are essential. So boron, for example, is required for some plants in limited amounts. Bromine is required for more marine organisms, and tungsten is needed for some microorganisms. They are not present usually in large quantities. So here you can see this is an interesting table. This is the amount of these metals, these transition metals that are present in an average human weighing about 70 kilograms. And you can see there's relatively small amounts of these metals in total. The most prominent one is iron. So you have roughly 4 grams of iron in you total, and a couple of grams of zinc, and then much, much less even of these other metals. So vanadium, chromium, manganese, cobalt, nickel, copper, and molybdenum. And you can see some of the roles that these play in terms of what particular enzymes they're present in, and to some extent what they do. And note the really predominant importance of iron in human biology. And so iron plays a role, essential role, for example, in iron storage and transport, in oxygen storage and transport, in transferring electrons to enable electron or energy storage, to enable reaction chemistry, etc. So most prominent within humans in terms of their importance are iron, copper, and zinc. And that also correlates with the amount that you have in your body. So our goal here is to really understand the various roles of transition metals within enzymes. And so let's first start out by defining what we mean by a metalloenzyme. So this is simply a protein that acts as a catalyst, but also contains one or more metal centers. And so important examples, some of which we'll consider, in detail are the nitrogenase enzyme, which does nitrogen fixation to form ammonia. There are examples that do oxygen evolution from water during photosynthesis and the conversion of oxygen to water during respiration. And so I realize most of you are familiar with proteins, but just to remind everybody what proteins are, and then we'll think about how the transition metals incorporate into these proteins. We're going to watch a short video, if I can get this to work. It's on YouTube. The link is here. And so let me try and bring this up so everybody can see. Okay, I'm going to play this. It's about 6 or 7 minutes. So it's actually quite a well done video and reasonably entertaining. But as I started, if you can't hear and see it, please let me know.
Proteins play countless roles throughout the biological world. Some transport nutrients throughout the body. Some help chemical reactions to happen at faster rates. Others build the structures that make up living things. Despite this wide range of functions, all proteins are made out of the same 21 building blocks, called amino acids. Amino acids are made of carbon, oxygen, nitrogen, and hydrogen atoms, and some contain sulfur atoms. Selenocysteine is the only standard amino acid that contains a selenium atom. These atoms form an amino group, a carboxyl group, and a side chain, all attached to a central carbon atom. The side chain determines an amino acid's properties and is the only part that varies from amino acid to amino acid. Hydrophobic amino acids have carbon-rich side chains, which don't interact well with water. Hydrophilic, or polar, amino acids interact well with water. Charged amino acids interact with oppositely charged amino acids or with other molecules. Primary structure. The primary structure of a protein is the linear sequence of amino acids as encoded by DNA. The amino acids in a protein are joined by peptide bonds, which link the amino group of one amino acid to the carboxyl group of another. A water molecule is released each time a peptide bond is formed. This linked series of carbon, nitrogen, and oxygen atoms is the protein backbone. Protein chains often fold into two types of secondary structures, alpha helices and beta sheets. An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl groups of nearby amino acids. Beta sheets are formed when hydrogen bonds stabilize two or more adjacent strands of amino acids. The tertiary structure of a protein is the three-dimensional shape of the protein chain. This shape is determined by the characteristics of the amino acids making up the chain. Many proteins form globular shapes with hydrophobic side chains sheltered on the inside, away from surrounding water molecules. Membrane-bound proteins have hydrophobic amino acids clustered together on their exteriors, so that hydrophobic side chains can interact with the lipids in the membrane. Charged amino acids allow proteins to interact with molecules that have complementary charges. The functions of many proteins rely upon their three-dimensional shapes. For example, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds to oxygen. Quaternary structure. Two or more polypeptide chains can come together to form one functional molecule with several subunits. The four subunits of hemoglobin cooperate so the complex can more easily pick up oxygen in the lungs and release it in the body. Different visual representations of proteins can give us visual clues about protein structure and function. This space-filling diagram shows all of the atoms that make up this protein. This representation, called a ribbon or cartoon diagram, shows the organization of the protein backbone and highlights alpha helices. This surface representation shows the areas in the protein that are accessible to water molecules. Most proteins are smaller than the wavelength of light. For example, the hemoglobin molecule is about 6.5 nanometers in size. Hemoglobin is found in high concentration in red blood cells. A typical red blood cell contains about 280 million hemoglobin molecules. The three-dimensional shapes of proteins determine their function. The flexible arms of antibodies protect us from disease by recognizing and binding to pathogens and targeting them for destruction by the immune system. The hormone insulin is a small, stable protein that can easily maintain its shape while traveling through the blood to regulate blood glucose level. Alpha amylase is an enzyme that begins digestion of starches in our saliva. The calcium pump is aided by magnesium and powered by ATP to move calcium ions back to the sarcoplasmic reticulum after each muscle contraction. Ferritin is a spherical protein with channels that allow iron atoms to enter and exit depending upon an organism's needs. On the inside, ferritin forms a hollow space with iron atoms attached to the inner wall. Ferritin stores iron in a non-toxic form. Collagen forms a strong triple helix that is used throughout the body for structural support. Collagen molecules can form elongated fibrils, which aggregate to form collagen fibers. This type of collagen is abundant in skin and tendons. Learn more about the functions and 3D structures of proteins and other molecular machines at the RCSB Protein Data Bank.
Okay, good. I hope everybody was able to see and follow that. Let me bring back the PowerPoint. Okay, so as you may have seen, there are a lot of different functions for proteins. So if you look within a cell, cells are really very chock full and crowded with proteins. And that provides lots of potential sites within these proteins where we might bind metal centers. And it's only certain amino acid residues of the polypeptide chains in proteins that provide strong ligands for binding these metals. And so the key amino acid residues for binding metals are shown here. And so we should think about the ligand properties of each of these. The first is histidine. So at the bottom of these structures, you can see this is the polypeptide chain, and here's the residue. And so histidine can coordinate to a metal through this nitrogen, which binds the metal. And it's important to note, as we'll see in a little later, this opposing proton on the imidazole ring, hydrogen bonding and pulling that proton away from this other nitrogen can actually attenuate the binding strength of the imidazole group. And so proteins use that trick to vary the strength of coordinating to a metal. Cysteine is going to have a deprotonated thiol, which gives thiolate or cystinate. And so this is an alkyl thiolate sulfur coordination. This, of course, will be a weak field ligand. Carboxylates are prominent in coordinating metals. So aspartate and glutamate have carboxylates that can coordinate to metals through these oxygens. Those also will be weak field ligands, of course, on the metal. And so part of the reason these are the more common metal coordinating residues is because some of these at least are deprotonated at physiological pH, which is about 7.4. And so we have a anionic ligand that can bind to the metal center. And it's not to say that other amino acid residues can't interact with metals. There are others that can and do. Generally, though, there's are weaker, significantly weaker binding interactions. So it's good to note that most proteins bind to metals as chelating ligands, where more than one amino acid residue is coordinating the metal. And so here's just an example of a manganese 2 site bound within the protein calprotectin. And you can see the manganese is coordinated actually by 6, in this case, different histidine nitrogens. So this here the protein is acting as a hexadentate chelating ligand. And as a result of the chelate effect, this binding of metals within proteins can be extremely strong. And so these proteins tend to really hold on to these metals and not let them go. And that can be very important for their function. You'll note that in the video you just watched, proteins are dynamic molecules, and they have some portions of them that move while they're being active. And so this brings to bear the point that these are multidentate ligands, but also they're flexible. And so usually there's flexibility of how these amino acid residues are arranged and how they can coordinate the metal. And proteins will use that tunability of the coordination sphere to tune then the either redox properties of the metal or reactivity of the metal center. Right? So this is a really important way that proteins can use metals. And so to bind these metals, the proteins will fold up into this specific tertiary structure. And if you have several of these metal binding residues in the same pocket, then that can be a strong binding site for a metal. But metals actually also sometimes help proteins fold into certain structures. So in the number of proteins, metal binding will have a large impact on the tertiary structure of the biomolecule. These metal sites can be bound in various parts of a protein. So this is just an example of a lactase enzyme. And you can see here 2 different types of copper sites. One shown in blue is a single copper ion. And that copper ion is located towards the periphery of the protein. And it's actually accessible to the water, the aqueous environment of the protein. And then the other type of site in this enzyme are these trinuclear copper sites, where you have three coppers strung together very close to each other. And these coppers in this site are actually buried within a hydrophobic pocket within the protein. And so that can limit this sort of pocket and surroundings of the metal sites can limit access by substrates, and is a way of the protein controlling what has access to the metals. Okay, so we said we'll see examples with of proteins that have even many more metals than this. And those metal sites, where they are in the protein structure, can matter a great deal for access to solvent, in this case water, substrate, molecule it's going to react with if it does, and also receiving electrons. So we'll see that proteins and enzymes have very specific electron transfer pathways set up, and where the metal is in the protein can either be along that path for electron transfer or not. You also have sometimes other ligands that are not part of the polypeptide scaffolding of the protein involved in forming an active site within an enzyme. So an example is shown here. This is the active site within a protein or an enzyme called soluble methane monooxygenase. And you can see it's bound by carboxylate and these imidazole ligands. It also has a water solvent ligand. And in this oxidized form, there's two hydroxide anions bridging between the iron centers. And then in the reduced form, you don't have those two hydroxide bridges, rather this carboxylate is the bridging species. And so it can be the case that you're required, that activity requires some additional species or ligands on the metal for activity. And so this is a very exciting and interesting metalloenzyme. So soluble methane monooxygenase actually catalyzes the conversion of methane to methanol under ambient conditions. And the way it does that is still not fully understood. But this would be a very technologically important reaction if we could do something similar. So converting natural gas into methanol, a readily transportable liquid fuel, is really an important research topic for many researchers. And right now we don't have very good technologies for doing this without quite a bit of energy input. But this enzyme is really set up to enable reactivity of this very inert hydrocarbon, methane, to give a specific product, just methanol. It doesn't overoxidize. You get out just methanol. And so understanding this could potentially lead to ways in which we might, on a larger scale, do this conversion. Another prominent bridging ligand that's found within certain enzymes is sulfur, and particularly iron sulfur clusters are very common in enzymes. And so you can see in these structures, the iron centers are tetrahedral in their coordination geometry. They have 4 sulfur ligands. So in this case, we've got 2 cystinate sulfur ligands and 2 bridging sulfido S2- anions. And the iron centers are, as I said, they're tetrahedral, and they're typically in an oxidation state of iron 2+ or iron 3+. So I'll ask a question then. Given these ligands, are these iron centers going to be high spin or low spin for their electron configuration? Anybody have an answer? Yeah, go ahead and speak up. Yeah, Amy, go ahead.
Is the weak field ligand, and iron is a first order transition metal, so I would say low spin.
So if you have a weak field ligand, does that enforce a low spin configuration? So a weak field ligand, remember, leads to rather small ligand field splitting energies. So the d orbitals are not very large in their splitting energy. So these are high spin electron configurations. So these iron 2, iron 3 tetrahedral sites, they're going to be high spin D5 or D6 electron configurations. Okay. All right, I hope everybody understands why these are high spin. In fact, for tetrahedral molecules, remember tetrahedral coordination gives a small energy splitting for the d orbitals, and most tetrahedral complex, almost all, are going to be high spin electron configurations. Okay, so here we have a number of examples of different coordination for an Fe2S2 dinuclear cluster. Some of these are Fe3S4 clusters, and sometimes, very commonly, you'll see an iron 4 sulfur 4 cluster, and these are what are called cubane clusters. And you can note that the arrangement of iron and sulfur atoms looks like a distorted cube. And in some enzymes, those cubanes are connected to a porphyrin heme group through a cystinate bridge. So these are examples of what are called cofactors. And so a cofactor is simply a non-protein component, meaning not part of the polypeptide scaffolding of the protein, but a component that's required for enzyme activity. So the enzyme really only does its function, does its reaction, catalyzed reaction, if that species is present. And so these iron sulfurs clusters are examples of some of the more prominent enzyme cofactors that contain transition metals. And these are used most commonly for electron transfer. So we'll see some examples of how these iron sulfur clusters can store and deliver electrons. But in some cases, they're also used for the reactivity of the metal and in small molecule activation in enzymes. Okay, so a really interesting and important example of an iron sulfur cluster cofactor is the iron molybdenum cofactor, which is found in nitrogenase. And so it has this very complicated structure shown here. So again, all of the iron centers are tetrahedral in their coordination. They're surrounded by, in this case, for this left-hand iron, you've got 4 sulfur ligands. For this these interior 6 iron centers, those are arranged in a trigonal prism. And at the center of the trigonal prism is a carbide anion, a carbido ligand, so C4 minus. And this is a really interesting and unusual species that's not commonly found in biology. And in fact, the presence of that carbon atom wasn't initially recognized in nitrogenases. And then at the right here, you've got another metal, a different metal, molybdenum, which is octahedrally coordinated by 3 sulfidoanions, histidine, and then this organic substrate. So as I said, this carbon atom was not initially thought to be present. In fact, it's difficult often to know the precise structure of some of the metal sites found within proteins. So proteins, as you know, are large molecules. That makes it difficult to determine with atomic precision the crystal structure of proteins. And some proteins are so large that this is virtually impossible. And the nitrogenase proteins, those, it was not that long ago that good crystals of these were obtained. And prior to that time, people really didn't know what these clusters looked like. They knew roughly the composition and spectral properties, but there was a huge research area devoted to trying to synthesize iron sulfur clusters that mimicked the spectral properties and composition of the cluster in nitrogenase. And so a huge amount of research went into understanding what this might look like before we had any direct structural information. And then actually, while I was a graduate student, that was a long time ago, let's see, that would have been, well, back in 92 or 3, crystal structures started to emerge of nitrogenase proteins. And the earliest crystal structures actually didn't have, this is a light atom here, so it doesn't scatter X-rays very strongly. But the earliest crystal structures were missing this central carbon atom. It wasn't there at all. They didn't know that there was an atom there. And so this led to people thinking there was 3-coordinate iron present in this form of the nitrogenase enzyme. And it led to a lot of synthetic chemistry of people trying to make analogs. But in fact, later, after more careful analysis and better data collection, it was discovered that there was a light atom in the middle. And initially it wasn't known whether that light atom was carbon, nitrogen, or oxygen. And for a long time, it was believed to be nitrogen as the most likely species. And so in the textbook that we gave you the chapter of, this is actually, at that time, they thought it was a nitrogen atom. But more recently, it's been shown and proven that this is in fact a carbon atom at the center of this molecule. Again, exactly how this works and doing its reaction is not entirely understood yet. But this enzyme catalyzes the reaction for fixing nitrogen and taking it from N2 to ammonia, which is essential as a fertilizer. And so this is a complicated reaction. It involves a single molecule of N2 reacting with 8 protons, 8 electrons, 16 magnesium ATP species to give 2 ammonia molecules, a molecule of hydrogen, a diphosphate, and monophosphate. If anybody has a question, please go ahead and speak up.
I have a question.
Sure, go.
How were they able to tell that it was a carbon in the end instead of
Oh, that's a great question. So let's see. So this was done first, initially, I believe, by a special form of electron paramagnetic resonance spectroscopy, where they could feed in labeled carbon and detect signals that confirm that carbon was present. And so the initial experiments, I think, were done by Brian Hoffman at Northwestern via spectroscopy. And those were just, it wasn't necessarily definitive proof, but it led to people really thinking it was probably carbon. And then later, other experiments started to confirm this, including some digestion studies and some even much higher resolution crystal structure determinations. Good question, though. There's actually a huge many-decade research effort to finally understanding the structure, and we're still trying to understand all aspects of exactly how the enzyme does this reaction. And in fact, it's again, a very useful reaction. This is converting nitrogen to ammonia is a reaction that we do on massive scales. We do it using the Haber-Bosch process, which is a highly optimized process catalyzed by iron. And it works exceptionally well, and it helps feed the world. But this process requires extreme temperatures and pressures, so 400 to 500 degrees C and 150 to 250 bar of pressure. Whereas this nitrogenase enzyme can actually do this reaction at room temperature under ambient conditions. So room temperature and 1 atmosphere of pressure. So this is, of course, of great interest, and a lot of research has gone into, can we make synthetic species that mimic this exciting reactivity? Okay, so another example of a cofactor where there's now even a much more substantial part of the active site that's not part of the polypeptide involves using hemes. So this is one example of a heme cofactor. And you probably saw in the video we watched earlier, certain enzymes have a specific binding pocket where the heme can slip into that pocket. And here the heme is coming in, and we're starting with iron 3, and it's 4 coordinates. So the iron 3 is down in the plane of the porphyrin to give this square planar coordination geometry. And so these hemes, as I said, they come in and can bind in a cleft in the tertiary structure of a protein. That binding is through a combination of non-covalent interactions, so van der Waals contacts, ionic interactions. But importantly, it's also through, or can be through, and is often through, adding a ligand to the iron center. So an amino acid residue often can bind on one side of the heme, providing a proximal ligand, making the iron 5 coordinate and changing dramatically its electronic properties. And so here we're going to talk about iron heme in enzymes. And so this is just showing some of the different structures that are found in heme containing proteins. And so usually these are going to be pentacoordinate at least, where you have you can have iron 3 binding an amino acid residue, either a thiolate, some cases a thioether, or a tyrosine, or a histidine. And then these are being set up. This ligand on the underside, as shown here, is generally termed the proximal ligand, and it's coming from a polypeptide amino acid residue. But that binding actually sets up reactivity on the other side of the porphyrin ring. And so that iron can then react with various substrates to form species such as shown here. And this will be the distal ligand, often where the reaction chemistry is happening. Right? So in hemoglobin, for example, you can bind O2 and form an iron superoxide species. And so this iron centers within hemes can convert between iron 3 and iron 2, depending on the availability of electrons and substrates and ligation. And this is just an example of how sublimely sensitive these are to ligation at this proximal site. So this is an active site in the enzyme ascorbate peroxidase, which contains a heme with this iron 4 oxo species. And we'll talk about this a little bit later in a different enzyme. But the reactivity of this oxygen atom at the distal site is very sensitive to this proximal axial ligand, right? And this is a histidine, but in this enzyme, the presence of this aspartate to hydrogen bond with this protein on the imidazole ring and pull it away from that nitrogen strengthens the binding of this other nitrogen for the imidazole ring to the iron. And so this residue, this aspartate residue, is essential for activity of the protein. In fact, this whole hydrogen bonding network is essential. You have to set this up to be in that position to hydrogen bond. And if you take out that residue, then this enzyme doesn't function. So these are all the clever tricks that nature has evolved to gain some really powerful reactivity, a number of reactions of which we're not very good at yet. All right, so one of the really key roles for transition metals in proteins is in electron transfer and bringing electrons for either reactivity or energy storage. And the electrons need to come in at the right places and at the right times. And all of that is orchestrated by the proteins. But this electron transfer generally most commonly involves iron 2+, 3+ redox couples, or copper 1+, 2+ redox couples. And it often can occur with electron transfer over long distances. And generally it's going to be an outer sphere electron transfer pathway where you don't have a covalent bridge necessarily connecting the metal centers between which the electron is transferred. And as I said, it happens in this very controlled fashion that involves molecular recognition to deliver electrons to particular sites at particular times. And I already mentioned this, but there are two common functions here. One is to deliver electrons at a certain step in a catalytic reaction. And the second is to deliver electrons in storing energy. or transferring energy as well. Okay, so let's look at a little bit at each of those possibilities. And the first will be involving this cytochrome P450 type enzyme. And these are very prominent in your liver for chewing up toxic molecules, and the kind of reaction that they perform very selectively is a reaction where 2 protons and 2 electrons plus a CH bond of an alkane and an oxygen molecule all react to form an alcohol and a water. So this gaining selectivity like this is really one of the great challenges of modern chemistry. But these enzymes do it very, very precisely. And so here are a couple of examples of reactions that different P450 enzymes can do. So here, taking camphor to this alcohol, or palmitoylic acid to this alcohol. It's a very specific site in these molecules that gets oxidized to the alcohol. And so the first question here is, we'll look at how these reactions happen later, but first question is, where do the electrons come from? Right? How do the molecules, how do these electrons get fed into the reaction? And a related question, which we're not going to get into in detail, but is equally important, is how are the protons delivered to this reaction site, right? And then, of course, how do substrates for the reaction enter? But for electron transfer, transition metals are essential often. And so here's an example of one of these enzymes. And you can see here there's 2 proteins. Here at the bottom is the P450 enzyme, and it has this heme cofactor as its active site, shown here in purple. Here's the cystinate bound iron center. And then this is at the top in green is a reductase protein. And this reductase protein is going to dock and bring with it an electron that'll be fed into the reaction to transform methane to methanol. And you'll note this reductase protein has an iron 2 S2 sulfur 2 cluster. And that cluster is located at the periphery of the protein, such that when it docks with the P450 enzyme, there's a relatively short distance for an electron to transfer from the iron 2 sulfur 2 cluster to the heme site in the protein, in the enzyme. And so this docking and everything in between is set up to facilitate that electron transfer. And the iron 2s2 is really serving to store and then deliver a reducing equivalent of electrons. So here's a little more detail on what that site looks like. But you can see the iron here, the electron here is going to go from one of these iron sites through this space of about 10 angstroms and wind up at the heme, right? So this is clearly an outer sphere type of electron transfer, but the pathway is very carefully coordinated to allow that electron transfer. And so just to take a look at the metal sites here. So before electron transfer, when the reductase protein comes in, we have 1 iron 3+ and 1 iron 2+. And after electron transfer, the electron transfers to this iron 3 cysteine complex. with the heme to form iron 2, right? And that iron 2 is then going to be used in doing the reaction to take methane to methanol. Okay. And any questions on that so far? If you have a question on what we've discussed so far, please go ahead and speak up. Okay, if not, let's continue. So next, let's think about electron transfer. And this will be electron transfer for energy storage in what's called complex 1. And so this is a key step in respiration. And here, this is very complicated, you can see, but here, this is a membrane bound protein that is at the top part of this diagram. And you can sort of just make out these orange spheres. These are iron sulfur clusters, and there's a whole string of them. And they're going to allow a cascade of electron transfer events to happen to inject an electron into this complex here. And this complex is then used for energy storage. So this is an incredibly balanced delivery system for electrons. And so the transfer here involves moving electrons over a distance of 95 angstroms as part of a catalytic reaction that's coupled to the pumping of protons across the membrane, right? And moving those protons across the membrane is the way that energy is stored. So here's a schematic view of that structure. And at the top, you can see this electron transfer protein. And so the orange circles are the iron sulfur clusters, and the blue pathway here is the electron transfer pathway. But essentially, these are strung along at gradually changing electron potentials, right? So to provide a driving force, you really have to have the potentials of each of these iron sulfur clusters poised so that the electron wants to move in one direction. And the spacing and intervening structures are set up to allow this sort of 10 angstrom electron hop between iron sulfur clusters. And then ultimately, the electron winds up reducing this molecule called ubiquinone, which is labeled Q here. And then that allows this electron to trigger proton pumping. All right, so you can see these are really elaborate uses for transition metals in setting up this cascade of electron transfers. And so the way nature tunes these potentials, the redox potentials for these metal sites, is of course using the ligand structure surrounding the unit, as well as its composition. And so here you see a plot just showing various different iron and copper containing proteins and their relative range of reduction potentials. And so this is the scale relative to the standard hydrogen electrode. And you can see it spans, over 1.5 volts in energy. And you can see the iron sulfur proteins, the potentials. You can have quite a variety of a volt different in potential just for iron sulfur proteins by changing the composition and ligation. And then those are generally reducing, for example, heme containing proteins. And then over to the right here are going to be the copper containing proteins. And so, as I said, these are all tuned by the composition of the cofactor and the ligands by the amino acid residues that bind the protein or bind the cofactor. Okay. So let's think about what's determining electron transfer rates here. So there are really 4 key factors, and they're related to things you've already learned about. Of course, the distance between the donor and acceptor are important, and the further away a acceptor is from its donor, the slower the reaction could be. But it depends a lot on the driving force. So delta G for this electron transfer reaction, as we saw with Marcus theory, though, this driving force can accelerate reactions up to a point, but then you could enter an inverted region where the electron transfer starts to slow. And so delta G has been optimized through evolution such that we can have these electron transfer cascades. Of course, what dictates this rate is really reorganization energy. And so nature has learned to tune coordination spheres and thereby tune reorganization energies. And that's also tuned by the nature of the intervening molecules and atoms. That is, how well are they able to facilitate the tunneling of this electron moving from a donor to an acceptor. So all of these things are key. And so reorganization energy, for example, generally you're going to see that those iron centers aren't changing spin state when they're involved in these electron transfers. So the iron 2 and iron 3 is high spin in those tetrahedral iron sulfur clusters. And so, not changing spin state helps minimize that reorganization energy. Okay, so let's think about and talk about enzymes and enzyme activity. So, of course, the role of the enzymes will be to lower the activation barrier for a chemical reaction. And this can be done by enzymes through a variety of means. So it could involve the binding of substrate, and that could mean transition state stabilization or ground state stabilization or orienting substrate appropriately for the reaction. And this can have something like 10 to the 8 rate acceleration in terms of lowering the barrier. And then proteins or enzymes often engage in catalyzing acid and base involved steps. And they can also involve covalent bond formation and help set up and lower activation barriers for covalent bond formation. And so all of this comes together then to, in some cases, very dramatically accelerate reaction rates. It could be, say, up to 10 to the 14 enhancement of a reaction rate relative to the non-enzymatic reaction. And so some of the critical factors to consider here for performing the reaction, we really have the 3-dimensional protein structure set up at the active site, so it's just the right shape with just the right functional groups and just the right positions to facilitate the reaction and lower the transition state relative to the reactant state. And another thing to really keep in mind is that these enzymes are dynamic. And so sometimes reactivity is actually very highly correlated with a motion. It could be quite a macroscopic motion of an enzyme. And that motion might, for example, bring substrate in the right place to receive an electron or to react with another substrate. And so there is another useful movie here that we can watch on how enzymes work, and it's going to give a specific example that really is well understood and very much highlights the complexity and beauty of enzyme reactions. So let me switch to YouTube. Where did it go? Okay, here we go.
Every single second, inside every living cell, thousands of chemical reactions are taking place. These reactions are performed by enzymes. An enzyme is a protein that catalyzes a chemical reaction. It initiates the reaction, speeds up the reaction's progress, and makes sure the outcome is always the same. These enzymes often work together to form longer pathways, such as the citric acid cycle, which is a series of chemical reactions used by cells to generate energy from carbohydrates. The essential tasks of life, such as metabolism, protein synthesis, and cell renewal and growth, are all regulated by enzymes. The life-sustaining power of enzymes lies in the fact that they catalyze reactions in mild conditions of pH, temperature, and atmospheric pressure. The rates of catalyzed reactions are millions to trillions times faster than those of the same reactions uncatalyzed. To speed up a reaction in the absence of enzyme, additional energy would need to be provided as heat, which jostles the substrates and occasionally provides enough energy to trigger a reaction. In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products. An enzyme acts as a template for the reaction, binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize the transition state, making it easier for the reaction to occur. To understand how enzymes work, let's take a closer look at a reaction in the citric acid cycle that is catalyzed by the enzyme aconitase. Aconitase binds to its substrate citrate and removes a hydroxyl group and a hydrogen atom to form the intermediate cis-aconitase. It then adds the hydrogen and hydroxyl back in slightly different positions to form the product isocitrate. In the active site, some amino acids are perfectly positioned to recognize the substrate and hold it in the optimal position for the reaction to begin. Some amino acids are involved in recognizing and holding the substrate. Other amino acids are directly involved in catalysis. Histidine 101 acts as an acid by donating its proton. Thanks to the chemical environment of serine 642, it can act as a base by accepting the proton from the substrate. The active site of aconitase also contains an iron-sulfur cluster that stabilizes the substrate electrostatically and helps to position it relative to the catalytic residues. The first step in the reaction is dehydration. In this step, histidine acts as an acid and protonates the hydroxyl on the substrate, allowing it to leave as a water molecule. Serine then acts as a base, extracting a hydrogen atom from the opposite side of the substrate, forming the intermediate cis-aconitate. Cis-aconitate then flips upside down, and the complementary hydration reaction is performed. In this step, histidine grabs a hydrogen atom from a passing water molecule, placing the resulting hydroxyl group back onto the substrate. Serine then returns its hydrogen atom, and the final product isocitrate is released. Notice that the enzyme itself was not changed by the reaction. It extracted a hydroxyl group and a hydrogen atom, and and then put them back, thus starting and ending in the same state. This is the hallmark of a catalyst. When it finishes a reaction, it is ready for the next, so it can perform thousands of reactions in a row. Notice also that the shape of an active site is often flexible. Many enzymes surround their substrates, enclosing them to form the perfect environment for a reaction. Enzymes are fundamental to life on Earth, working every second of every day to maintain life processes in every cell in every living creature on the planet.
Okay, let me go back to PowerPoint here. All right, so I think that video really nicely makes clear how concerted and very specific to a particular substrate these enzyme reactions can be. And so carrying out that sort of very concerted, highly selective transformation is a very difficult thing for synthetic chemistry to do in the same very selective, very precise way. So just for metalloenzymes, a few considerations. Their metals in enzymes are going to have all the same types of mechanisms for accelerating rate as other enzymes, the ones we just went through. They can facilitate also, though, quite readily and with quite a bit of precision and range on the voltage, oxidation and reduction of substrates. And that can enable reactivity that's often difficult without using metals. And they can also allow breaking and forming of particularly strong bonds. And that usually is simultaneous with electron transfer and proton transfer. And so examples are breaking the OO double bond of oxygen, as in cytochrome C oxidase in respiration, forming O2 double bond. So here you're forming a bond, taking oxygen atoms from water in photosystem 2 during photosynthesis. And as we saw, breaking the very strong triple bond in N2 to form ammonia in the nitrogenases. And so all of those, really can't readily be done without using transition metals. So let's take, finally, we're going to take a look at how a cytochrome P450 enzyme operates, some of the details of its reaction steps. And so these are drugs, these are enzymes that can break down drugs in our liver, and they're responsible for about 75% of drug metabolism. They catalyze synthetically relevant activation of an aliphatic CH bond using O2 as an oxidant, and they're particularly well understood mechanistically. And yet still we struggle to recreate this sort of reaction in synthetic systems. And so here is the sort of reaction and electron transfer sequence that sets up the site that's called compound 1 in a P450 enzyme that's thought to be responsible, the active species that does the transformation of methane to methanol. And so you can see this is a very involved reaction sequence involving reduction to iron 2, binding of oxygen to form an iron 3 superoxide, an additional electron transfer step, proton transfer, guided protons to form this hydrogenated species. And then ultimately, this compound 1 has the form where you've got an iron 4+ formal oxidation state with an oxo, so an iron 4 oxo species, multiple bond between metal and ligand. So this, of course, is within the heme unit. The heme is actually a radical cation. And you've got this cystinate ligand on the proximal side of the heme, right? And so this is where the reaction is going to occur. And so this is really an amazing reaction sequence. But here's the step where you're actually converting an alkane. So the alkane comes in, this very reactive iron 4 oxo species, which normally would react with many different species. It's protected within the protein pocket, and that protein environment delivers an alkane or an aliphatic CH bond containing substrate. And that will, this iron oxo is set up then to cleave the RH bond, and that creates a hydroxyl species and a radical. And there's then a rebound. So the rebound step here involves this radical not escaping the protein pocket, reacting now with the OH and forming an alcohol, which is released. And that then comes back to the ferric resting state to allow another oxygen to come in and form a new compound 1. And so one of the amazing things about how this is done is, first of all, it enables under ambient conditions this reaction to proceed very rapidly. And that's in part because it happens rapidly and without termination, in part because the protein environment keeps, oops, I'm not sure what happened there, keeps this radical species from diffusing away, right? If this radical could escape, it could react with all kinds of things. But the pocket within the enzyme keeps that radical positioned such that it then rebounds, reacts with the hydroxyl to make the alcohol. And once it makes the alcohol, that alcohol immediately wants to leave the reaction pocket. And it doesn't undergo a second reaction with incoming oxygen. And so the key here is that it has a long-lived reactivity because the radical doesn't escape, and the substrate doesn't get over-oxidized. So this alcohol doesn't get further oxidized. Other CH bonds aren't turned into oxidized functional groups in this reaction. Okay, so I think that's it for today. We have time for a question or two. Anybody have a question?
Hi, Professor. So I have a question on the topic of cytochrome P450 enzymes.
Mm-hmm.
So in my previous toxicology class, I think the course presented cytochrome P450 enzymes as rather non-selective, in the sense that they could often cause bioactivation. So I was wondering whether that was an oversimplification or whether there was some other explanation.
It depends on what you mean by selective. And so, yeah, actually, so the selectivity here it's attacking CH bonds in particular and forming an alcohol. And that will leave, but then other, it works on many different CH bonds in many different molecules. So the specific reaction sequence here is very selective, but it works on lots of different substrates. So here, I guess I used selectivity to mean this is the reaction path per cycle.
Got it. Thank you.
But you're right. You're absolutely right. So these are particularly good for breaking down all kinds of aliphatic CH bond containing molecules in the liver. Good question. Other questions? Okay. If not, on Thursday, we'll reconvene and we'll start discussing F element chemistry. All right. See everybody then.